Good morning, everybody. Thanks to CENIEH for hosting this event, and to Emiliano and the organizers for inviting me. And I look forward to a good conference with the other participants, and I really look forward to learning a lot from these sessions. I think that Emiliano may have made a mistake in inviting me to a conference on human paleoneurology, because that suggests the study of the fossil record of human brain evolution, paleoneurology, and in fact I don't study fossils whatsoever. I study the brains of living species, and I make comparisons among humans and our close relatives to try to understand what are some of the key variables that have led to the evolution of our cognition. But I will do my best to represent my work in the context and the framework of paleoneurology and highlight the connections that can be made between the study of comparative neuroscience, the sorts of research that I participate in, and the field of paleoneurology. So I am fundamentally an evolutionary anthropologist and a quantitative neuroanatomist. And my research focuses on comparing brain structure of humans and other mammals with a particular focus on great apes. The main aim of my research, and probably the goal that unites all of the participants in this conference, is to understand the human brain's place in nature. To try to determine what might be unique about our brain organization, what's shared in common with our close relatives, both the living great apes, chimpanzees, bonobos, gorillas, and orangutans, as well as fossil hominin ancestors. We're going to hear a lot more about the fossil hominin ancestors in the coming days. In this talk, I'm going to be presenting research from my lab concerning comparative brain anatomy, pertaining to what might be unique evolutionary specializations in humans, in comparison especially to great apes. So as our closest living relatives, chimpanzees and other great apes resemble us in many ways. Our lineages split from one another about 6 to 8 million years ago. But despite a large number of similarities in our phenotype in terms of anatomy and behavior and cognition, the human psychological specialization is especially distinctive. And we look to neural changes that might correlate with human cognitive evolution. So what you see here is a list of proposed cognitive specializations in modern humans, offered by Peter Carruthers, who's a philosopher, in his book, The Architecture of the Mind. And this, I applaud him for coming up with exactly 22 human psychological specializations, and this is something that people can certainly debate. But this does highlight some of the uniqueness of our modern human way of thinking, involving things like the evolution of language, enhanced representation and abilities of the mental states of other individuals, as well as a better understanding of our own mental states, being able to project our minds into the past and the future and consider different options and kind of rehearse different mental scenarios of ourselves. A lot of people also emphasize enhancements of empathy, social cognition, and our ability to cooperate with one another. More than half of these putative human psychological specializations are on display in this wonderful movie, The Departed, Academy Award-winning Best Picture from 2006, directed by Martin Scorsese, involving an undercover police officer from Massachusetts infiltrating the Irish Mafia, and in turn someone from the Irish Mafia infiltrating the Massachusetts police. And it's just a nice example of all of the kind of work that the human mind can do in understanding other minds, and in deploying creative thinking and language, in this case for deception. But oftentimes, also for the purpose of prosocial ends like cooperation. So, as we try to understand the emergence of these kinds of human psychological specializations, we look to the neuromachinery that drives cognition, and that is, of course, changes in the brain. And we can do this, this is the field of human evolutionary neuroscience, from two different perspectives primarily. First, there is paleoneurology, which is the study of human evolutionary neuroscience that focuses on changes that can be seen in the fossil record, from studying the impressions left by brains and the associated soft tissues as they impinge on the hard tissue, the intertable of bone in the skull. You're going to hear a lot more about that. Paleoneurology provides a perspective on the timing of certain changes in the brain, and it has the advantage of allowing us to link what can be seen from neural structure, from its morphology, its shape, and what can be gleaned from those endocasts, to connect with evidence, other evidence from the record of the past, evidence for changes in archaeology, climate record, geological change. So it provides direct evidence of what brains were like in the past, to some extent, and how that might relate to other changes in the past record. But paleoneurology is very limited in the kind of information that it can provide. And I think we all know that. You can observe the surfaces of brains, much like what you see in this representation of a human. This is a side view, a lateral view of a human, modern human endocranial cast, digitally rendered, and a modern chimpanzee endocranial cast. And you see the general impression of the shape of the brain, and some of the folding impressions on the surface, and some of the blood vessels that pass over the surface of the brain, but you're missing quite a lot of information as well. Naturally, you're missing all of the detailed record of how the brains fold. On the cortical surface, you're missing information about internal structures that might only be viewed from MRI, like the volume of a structure deep inside the brain, such as the caudate nucleus or the thalamus, for example, which could be seen with MRI in modern brain specimens. You lose information about cellular morphology, different cell types, synaptic connections, the pattern of white matter connectivity in the brain, molecular expression, all sorts of biological variables that we know from a huge amount of neuroscience telling us that these are variables that influence cognition and behavior. These cannot be dismissed. Certainly brain shape is one variable, but we know from clinical pathological correlations, gene linkage studies, evidence from neuroimaging, and from many model animal studies that these variables make a difference. You can think about, for example, variants, polymorphisms in serotonin transporter in humans, which relates to individual variation in risk-taking behavior. And this is certainly the sort of thing that is a salient evolutionary behavioral characteristic, which would not necessarily be evident from paleoneurology. So we can also get information by doing comparative neuroscience, not just from more details of brain shape, but also cellular architecture. What you see here is a silver-impregnated neuron from the prefrontal cortex of a human. And you can see all the detailed morphology of the dendritic branching and the spines, which are receptive ends for synaptic connections. So I was encouraged in the early days of my research by my advisor, who was a paleoneurologist, a man named Ralph Holloway. I was encouraged by him actually to pursue studies of comparative neuroscience because it is a more data-rich avenue of research. And also, as he pointed out to me, and as I soon learned, it's tremendous what we don't know about the basic comparative neurobiology of humans relative to apes. There's been extraordinarily little study of
the detailed anatomy of the brains of great apes that allow us to see in comparative relief what is distinctive in humans and what might be shared. So a paleoneurologist set me on my career as a comparative neuroscientist. Now, of course, the history of comparative neuroscience is storied and goes back to the early days of our understanding of Darwinian evolution. Before there was very much of a fossil record to study for paleoneurology, when only some Neanderthal specimens were known from Belgium, Gibraltar, Neander Valley, Charles Darwin published his major work on the origin of species, leading to the idea that there is continuity in the tree of life. Humans are related to other organisms, particularly primates. And what erupted in consequence after Darwin's publication was a debate which took place in the public sphere to some extent between Thomas Henry Huxley, a staunch defender of Darwin's ideas, and Sir Richard Owen, who was a very well-regarded comparative anatomist and paleontologist. He was the head of the natural history collections in the British Museum, but he was a critic of Darwin's idea that humans evolved by descent from a common ancestor shared with apes. Richard Owen instead believed that humans were so different and special that they should be considered part of an entirely separate taxonomic category from other species. T.H. Huxley, in contrast, argued in favor of Darwin's view of continuity, and a very important piece of evidence in this debate concerned comparative brain anatomy. So before there was neurological evidence, there was comparative neuroscience that entered the debate. So in support of his view, Richard Owen claimed that an anatomical feature buried deep internally inside the lateral ventricle of the brain, called the hippocampus minor, which you would see in this dissection. This is a top-down view with a reflected part of the superior cerebral cortex removed. And there's a bump, which was called the hippocampus minor. And Owen argued that this was only present in humans and therefore represented a discontinuity from the rest of the natural world and favored the perspective that humans are special and unique. And in contrast, T.H. Huxley did very careful anatomical dissections of many more specimens and found that there was a detectable hippocampus minor in other apes, favoring for the view of continuity and gradualism. So, in fact, Darwin invited Huxley to contribute an appendix in the second edition of The Descent of Man, in which Huxley reviews in great detail the comparative anatomy of the brain in humans and apes. And clearly, Huxley is arguing in this passage for gradualism, continuity, and brain structures between humans and other apes as evidence of Darwinian evolution. And in fact, he's correct. The human brain does not have any obvious new parts, like a hippocampus minor. This is what we now know from many, many more years of research. There are no new parts. And the differences between human brains and ape brains are of the sort described by Huxley. They do not constitute a sharp demarcation, but rather are resemblance of fundamental characteristics and more subtle, perhaps, quantitative sorts of differences. Differences of degree, but not in kind. And that's, I guess, where I come in as a quantitative neuroanatomist, because many of those differences between human brains and ape brains, which will relate to our species' unique cognitive specializations, do come from those differences of degree rather than kind, and those have to be quantified. So, when looking in the brain for evidence of the neural basis of extreme behavioral specializations, perhaps in humans something like language or our capacity in mental state attribution, in looking at other species, you can find some pretty dramatic
cases of brain reorganization, particularly in the neocortex. And think of some classic examples like the somatosensory representation of the bill in platypus, or the star-nosed mole with its elongated sensory apparatus in the snout. And you can see here shown in black on a tangential preparation of the cerebral cortex surface, the enormous size devoted to representing the specialized sensory functions in these brains. And this really corresponds to what Jerison described as the principle of proper mass. The more important a behavior or structure is in the ecological social adaptations of organisms, the greater amount of brain tissue devoted to the function. We can see that pretty well represented. So it leads to the obvious question: in humans, then, do we see anything akin to this type of cortical reorganization? So in the human brain, what is the evidence for this type of reorganization? And Darwin knew it, and it's fairly obvious, hard to ignore. One of the most dramatically different things about the human brain, which must in some way correspond to these adaptations, is a tremendous enlargement of one part of the brain called the neocortex. And the neocortex in humans is about 3 times larger than what we would expect for a primate of our body size. At around, on average, 1,400 grams, it is the largest brain in primates, and the neocortex comprises most of the brain. We know from the paleoneurological record that most of this tripling in brain size and correspondingly neocortex size occurred within roughly the last 2.5 to 3 million years of human evolution. So, human neocortex is large. It's the most disproportionately enlarged part of the brain. What is the neocortex? I'm going to focus on this part of the brain because it is the region of the brain most important for processing a lot of these psychological specializations that we consider uniquely human, like language. What you can see here is just a gross view of the outer surface of a human brain compared to a chimpanzee brain. And the neocortex is what's seen as this wrinkled surface on the outside. Now, looking at the neocortex in cross-section of a gross human brain specimen, here it is cut roughly coronally through the middle of the brain, and you can see some of those internal structures I referred to before, like the thalamus, which would not be evident from paleoneurology. And you can see that there's this gray matter on the surface of the neocortex and an underlying white matter. Together, the gray matter, which contains the cell bodies, the white matter, which contains the axon connections, comprises about 80% of total brain volume in humans. The neocortex architecture can be appreciated under a microscope, and it has a very distinctive pattern of cellular architecture. So if we look at this piece of neocortex zoomed in, it's about the size of a pinky nail. And here we see a comparison of architecture of the neocortex in 3 different primates: humans, chimpanzee, and macaque monkey. And despite a 15-fold variation among these species in overall brain size and overall neocortical size, you can appreciate that neocortex thickness doesn't differ very much across these species. And in fact, there's very similar numbers of neurons in a vertical array of cells within the neocortical sheath across many, many mammals. There's some variation, but it's not nearly as significant as the variation in the tangential surface area of the neocortex. And so this architecture can be studied. And Neuroscientists have recognized 6 different layers in the neocortex based on cell size and packing density. And these regular differences in the cell architecture then can be used as a guide to compare regions within the same brain and also regions across brains. These 6 different layers have a very fundamental stereotypical organization, which is also useful in drawing inferences about function just from looking at anatomy. I'm not going to get into the details of this, but in general, more superficial cortical layers house neurons that make association connections between different cortical areas, and deeper cortical layers house neurons that make output projections to regions like the cerebellum and the spinal cord. And the middle layer, in layer 4, receives input projections from the thalamus, incoming information to the cortex. And then there's a stereotypical pattern of connections within a vertical array of cells called a cortical column, which can be recognized. So using neocortical architecture then, one can observe differences across the cortical sheet, even within single individuals. So these are a couple of images from an orangutan brain, and you have primary motor cortex on one side and primary visual cortex on the other side, and you can appreciate there's great difference in the thickness of the cortex and also the cell packing. So in primary motor cortex, you don't have much incoming sensory information. It's mostly involved in motor control, so you have very large output neurons in layer 5, BET cells. In primary visual cortex, you have, in primates, since visual sense is so important, you have an expansion of processing within the input layer for thalamic inputs, layer 4. And these anatomical differences that can be recognized with the microscope and histological preparations have a correspondence to functional differences. So you can see the boundaries of primary motor cortex under the microscope from brain sections in many, many different species. There's very well recognized homology. And in fact, like I mentioned before, there are no new areas in the human brain that have been recognized, that have been identified so far. So even in the neocortex, even in the brain that subserves unique functions like human language, we think that those functions are built on a more ancient layout of cortical regions. So here you see a comparison of maps of the dorsolateral frontal lobe by looking at that kind of cell packing architecture, comparing macaques and humans, and also these sorts of maps are supplemented by data from connectivity experiments. And you see that there's very good homology and correspondence from macaques to humans, even including parts of the frontal cortex like rodents area 44 and 45, which are language. We can see these homologies as well between humans and chimpanzees. Okay, so now that I've set the stage for understanding the approach from comparative neuroanatomy, I'm about to dive into some of the research findings from my lab. And at this point, I really want to be sure to acknowledge and recognize the fact that all of this research is a team effort and involves the very hard work and creativity and sweat of many students, postdocs, and research assistants. I'm here really just as an ambassador or as a spokesperson for the research in my lab. I want to be sure to recognize I'm here, and then I'll also point out as we go through some of the data who's actually been doing the research, because in many cases it's not me. I might have had involvement with the ideas and writing it up, but we all know with lab-based science that these are team efforts, and I want to recognize that. And so you may recognize someone sitting in the front row from my lab meeting, Aida Gomez-Robles, who comes from this very spot in Burgos, CENIEH, and she joined my lab approximately 6-7 months ago. And towards the end of the talk, I'll present some of her research from the lab. Okay, so
what I'm going to tell you about now is some research that we've done in mapping cortical territories across species. And this is work that was completed by my PhD student, Amy Barnfind, looking at the parcellation and the volume differences in regions of the insular cortex in primates. And the insular cortex, you can see here, is buried underneath the overhanging frontal, parietal, and temporal lobes. So this is a part of the cortical surface that cannot be seen from endocast because it's buried under surface cortical structures. And in humans, we know very well from neuroimaging studies that there's a gradient in function going from posterior towards anterior in the insular cortex. More posterior regions here indicated by this red arrow are involved in the sensory representation of visceral body states, monitoring the state of your viscera. It receives spinothalamic inputs from small diameter axon fibers that convey pain and temperature sense as well. But then as you move more anteriorly, progressively more anterior in the insular cortex, it's as though the brain takes that body state sensory representation and adds onto it, including more information about a conscious awareness of one's own body state, so sense of disgust and hunger, or localized more frontal regions or more anterior regions of the insular cortex, but also very importantly, social awareness of body states. So one's ability to understand other people's emotions by reference to one's own body states. The ability to have empathy for other individuals seems to be localized to the more anterior regions of insular cortex. And we know that there are certain diseases like frontotemporal dementia, the behavioral variant of frontotemporal dementia, which particularly affect frontoinsular cortex, wiping out neurons in this region and causing widespread degeneration. And some of the symptoms of behavioral variant frontotemporal dementia include a patient's inability to recognize the emotional impact of their actions on others. So these patients will start to say very rude things without knowing that they're being rude. They'll say, "Oh, you look very fat today," just very matter-of-factly. They lose their sense of disgust. So you'll see videos of these patients picking up half-empty bottles of soda from the trash can and just drinking with absolutely no shame, no disgust. So these social emotions are lost and seem to be very associated with the function of anterior insular cortex. So this might be a very important part of the brain to study in understanding the evolution of human social cognition. So this is a region of the brain that also shows histological cytoarchitectonic parcellation in a posterior to anterior gradient. And you can see that it's been parcellated based on the granularity or the thickness of layer 4 in the cortex, moving from posterior to anterior. And this has been done showing homology in humans, macaque monkeys, and also some other stretch-like primates. So what Amy Barnfind did in her research is use this sort of information in the homology to look at brain sections from many different species of primates to study changes in the volume of regions of the insular cortex. And I'll point out that also there's such an emphasis on the function of the frontoinsular cortex in humans that, and apes, that there appears to be the evolution of a unique neuron morphology in this part of the brain, as well as the anterior cingulate cortex. It's called von neurons. You can see in the left side image, there are these very long, slender projection neurons which don't have the typical pyramidal-shaped morphology of other projection neurons in the cortex. And they're also found in other large brain social mammalian species like elephants and whales, so they might have some role in conveying outputs of these social emotional circuits in large brains, given the geometric constraints of large brains. But they're also found in much smaller numbers in other mammals, many other mammals. So they're an interesting neurotype and a topic of much further study. So using cytoarchitectonic differences across many different primates, then
you can start to parcellate these regions of the insular cortex. This is what Amy did. We did this from brain sections of 30 species of primates. And this is from our own slide collections and some existing slide collections. It's very hard to find whole brain slide preparations of primate brains, and most of them, I should disclose, do come from zoos and biomedical facilities, from animals that have died natural deaths in those conditions. Okay, so with the data then, looking at species means, we regress the volumes of each of these components of the insular cortex against the rest of the insula and against brain volume. And what I want to draw your attention to from these data, which I don't know if those of you in the back of the room can see very clearly, but as we move from B to C to D to E, which represents more and more anterior regions of the insula, the slope of the regression increases. So it goes from nearly isometry to more and more positive allometry, okay? Significantly positive allometry. And this is based on both contemporary species means as well as phylogenetic least squares regression. And so this indicates to us that there is hyperallometric scaling, a disproportionate increase in the anterior parts of the insular cortex. So that machinery for social cognition, with brain enlargement, a byproduct seems to be that it just gets disproportionately larger. And this might in part help to explain the very strong correlation between overall brain size or overall neocortical size and things like social group size across primates. One other thing I'll point out is that the human point, now here indicated in red, is right on the line. So even though we appear to have some psychological specializations related to social cognition in terms of empathy, cooperation, we have exactly the amount of anterior insular cortex. Indeed, exactly the amount of all insular cortex regions that we ought to have for a primate of our brain size. However, what this means is that for having such a large brain size, which is about 3 times larger than we would predict for other primates, that buys for us as a byproduct a greater share of representation of anterior insula. So it's interesting, but not exceptional for humans. Now, those sorts of scaling studies, I think, are really essential to understand the size and proportions of cortical territories in humans in evolutionary context. Without doing those broad comparative studies and looking at scaling, you can't always decipher what is the product of expected allometric enlargement versus what's a departure. But that's not always practical in comparative neuroscience. Many cortical territories are very difficult to define. It's very labor intensive, it takes a long time. And so what I'm showing you here instead is a table of all of the data that currently exists, making the narrow comparison of human cortical region size relative to humans and bonobos. So our closest living relatives, and this is not in the context of scaling, but just the fold difference in human cortical territory size versus these other species. And this is a very limited set of regions. Out of a possible around 150 cortical areas, we only have data from a small number of regions. And what I'm going to point out, I might have to step away from the mic to be able to point, unless there's a laser pointer somewhere, is that if we consider our standard for fold difference to be the overall brain or neocortical gray matter, that would be around 4-fold. So any cortical region that's 4 times bigger is simply different between humans and chimps according to the same amount as the total neocortex. Anything less than that means that it's proportionally smaller, anything bigger proportionally larger. And what you'll notice is that of all the regions that have been measured, the proportionally largest ones are all in the prefrontal cortex. Now this is, and Emiliano would be interested to know this is in part because we haven't measured any parietal areas or very few temporal areas.
those areas are harder to define. The homologies have not been established as clearly in primates, so it's a more intractable question. But prefrontal cortex seems to be disproportionately enlarged in humans as compared to other regions, like primary motor cortex, primary visual cortex, which indeed don't even keep pace with overall neocortical enlargement. So this suggests that association areas of the neocortex, higher order cognitive association areas, have increased disproportionately in humans compared to our close relatives, chimpanzees. Now, I want to point out, and having the bonobo data from a few regions helps me make this point. In comparative neuroscience, very often we fall into the trap of treating common chimpanzees, where a species where brains are more abundantly available for research, we treat them like a stand-in for the last common ancestor of humans and chimps. And we look at these human-chimp differences and assume that changes in humans are recently evolved and that the chimpanzee brain is static evolutionarily. And that's absolutely not true. And you can see this when you also make the comparison of cortical area fold differences between humans and bonobos. And there's some very large differences between the results you get when you look at bonobos and when you look at chimpanzees. And that's because bonobo brains and chimpanzee brains are different from each other. And there's more and more evidence to point that out. And this is what we'd expect exactly, because they're very different behaviors expressed by these two plant species. Okay, so what you see in the upper panel is just a color table of the fold difference in cortical area volume between humans and chimpanzees from what I showed you. What's interesting to me is the way that it corresponds for the areas that have been studied very closely with the kind of data that come out of David Van Essen's lab, looking at the cortical deformation between humans and macaque monkeys from a landmark-based approach. And you can see also that in that comparison, it's the association areas of the prefrontal cortex, parietal cortex, and temporal cortex that are disproportionately enlarged in humans. And then similarly, in development, in this lower part of figure C, also from David Van Essen's lab, showing the difference in cortical expansion from newborn humans to adult humans. And similarly, it's the association areas that change the most in volume from newborns to adult humans. And so this suggests that there might be a general effect of cortical territory allocation when you have an enlargement of the cortical sheet. There are important organizing growth centers, which are located primarily in sensory regions, and diffusion gradients away from those sensory areas might lead to, with selection for an increasing cortical sheet, might lead predictably to the expansion of association areas. So again, here is a manner in which humans, it helps us to understand the natural history of human cortical organization, but in this regard, perhaps we're not exceptional. Okay, so besides changes in the size of cortical region volumes, their internal histological organization might also differ among species. So the details of the wiring within cortical areas and among cortical areas certainly important for driving psychological differences between species. And none of these sorts of species differences, of course, can be seen from paleoneurology evidence. So in my lab, we've been also studying how the histology of the microstructural anatomy of cortical areas differs between humans and other primates. And the data I'm going to show right now, the research was led by a postdoctoral scientist who used to be in my lab named Mohammed Spakter. And what we studied was the neuropil fraction in cortical areas, and that is the spacing between cell bodies, which is occupied by connectivity. And this is just a quick and dirty way of estimating. Here you see a comparison of humans and chimpanzees for 6 cortical areas, and we studied 12 adult individuals from each species, both hemispheres. And they include regions of the prefrontal cortex, regions that are important parts of Broca's area for speech production and comprehension, language, as well as Wernicke's area homologue, primary auditory cortex, and primary motor cortex. So a nice collection of different areas from both hemispheres. And you see from the histology just the cell body stain. And I'm going to zoom in on the primary motor cortex of a chimpanzee. And we collected a systematic random series of image frames from each region of interest. This is what a single image frame looks like. We did this in a number of sections through each region. Each image frame is then converted to binary, and we calculate the percentage of the frame that contains darkly stained cells, the black space, versus the white space, or the unstained portion, which is occupied by neuropil. And that neuropil space is everything that is not cell body. So that includes dendrites, synapses, microvasculature, and